Welcome to the Genetics Podcast. I'm Patrick, the co-founder and CEO of Sano Genetics, and I'm really excited to be here with a guest that I've been hoping to have on the podcast for a very long time, and that's Dr. Matt Might. The reason I'm so excited to have Matt on the podcast is because he has an incredible personal story and has inspired many others to go into research in rare disease. So I'm not going to spoil too much by giving the long intro to Matt, but I'd just like to say welcome, Matt, and it's great to have you here on the podcast.
Well, thanks. It's a pleasure.
Great. So I was wondering if you could just start by telling us how you got into rare disease from personal perspective with your son and subsequent blog post, Hunting Down My Son's Killer, that really sparked probably a lot more than you imagined when you wrote it.
Sure, I can sort of recap up to that point. Well, yeah, for starters, I'm not a biologist or anything like that by training. My training is all in computer science, and still have spent most of my academic career doing just computer science. These days it's medicine, but the reason for the shift is in fact my son. So I have a 12-year-old son, Bertrand, who started off his life with a 4-year diagnostic odyssey where we had no idea what was wrong. He had a movement disorder, he had seizures, he had developmental delay, and he had this curious lack of tears linked to all sorts of problems with his eyes. And we had no clue why that was, again, for the first 4 years of his life. Ultimately, that diagnostic odyssey culminated in one of the first human trials of exome sequencing for these intractable diagnostic odysseys. And that was done at Duke. To our delight, they found an answer. And to our surprise, they told us that he was, in fact, the first patient they'd ever seen with this brand new disease, which is called NGLY1 deficiency. So he was missing, essentially had, a loss of function in both copies of his NGLY1 gene. And so, they believed that they could tie this up to the high-level phenotype. And, that he was the only one they knew of. There was no record of this disease anywhere in the literature, no other patients known, no prognosis. And at that point, they said, so it's really kind of on you. And I really ran with that. So I really knew I had to do two things. One, I said, we have to find other patients, and they must be out there somewhere.
Right.
You could even compute roughly how many there should be based on the frequency of the likely pathogenic alleles there were in the population genomics databases at the time. And secondly, I said, we've got to do some science to figure this thing out and figure out how to treat it. So the blog post came in that, I needed to find other patients. And so I wrote a blog post called Hunting Down My Son's Killer that was very deliberately designed to do two things.
Right.
One of which was to go viral, and you can probably figure that out from the title. And there's a picture of Liam Neeson holding a gun up at the top to help out with that too. And the next thing was wanted to rank very highly in Google search results for the kinds of terms that another patient or their parent was likely to type in. So things like lack of tears or why doesn't my child cry? I wanted it to appear for those kinds of searches so that other people with this disease would start to find us. And that is exactly what happened. So that brings us to the end of the diagnostic odyssey and the formation of the community.
That's amazing that you actually approached it. I wouldn't have thought about the foresight of writing it virally, doing the search engine optimization, but it makes complete sense. You think that was your computer science background, or what caused you to think about it in that way?
Yeah, I think it was very natural for me to think of it that way as a computer scientist. I mean, I know how Google works. I know how it thinks. And so, that's exactly what search engine optimization is all about, is how do you trick Google into ranking your post higher? And I'd been blogging for a long time at that point. So I knew some of the basics of that, just from writing my own personal academic blog.
How many families got in touch with you as a result of the blog post? I can see from the edit history of the blog post that it goes from 2 to 8 to 15. And I think today something like over 60 families that have been diagnosed.
It's definitely over 60. So I really need to go up and go back and edit that blog post a bit to reflect reality. In fact, we'll be close to 70 at this point. I bet we are. I haven't checked the total count recently, but it's been a lot.
That is incredibly rare, though, on the scale of rare disorders, isn't it? I mean, how did that square with your calculations originally when you look at the population genetics databases? Was it clear that there might be hundreds to thousands?
I estimate we're in the mid hundreds, so probably 500 or so patients out there. So that to me more or less squares with those calculations. It's just to say, yeah, it's very, very rare.
Very rare.
To me, it's remarkable that we found that many of them.
Why is it so rare? Is it a very small gene? Is having one copy also not doesn't lead to full health, or why is it so rare?
It's a good question. I don't know why it's so rare, but actually, since you're asking the question, I just realized I've never actually checked to see what the metrics for haploinsufficiency were on Nomad. Sure enough, there doesn't appear to be any adverse impact on health, at least prior to reproductive age, according to the estimates on Nomad. So I don't know why there aren't more out there. But certainly the loss of function alleles are rare enough that we don't get a lot of patients.
Right. I just was looking it up. To people who don't know what Nomad is, it's a database of largely healthy people, and it can be used in the way that the analogy that's often used is when fighter jets go to war, the ones that come back, you look at where the bullets are, and the ones that don't come back, they've hit the fuselage or whatever. So you can use the ones that come back to figure out the quote unquote weak points of the plane. And there's a similar idea in human genetics where you can look at large numbers of quote unquote healthy people and look for genes where you don't see genetic variants in it. And it looks like, just at a quick glance, that NGLY1 is not particularly high PLI, meaning it's not an issue, as you said, to have people knocked out for the gene in the population, but having both copies knocked out, as you know, is not compatible with the healthy life. So it's interesting. I mean, I guess it just goes to show there's 20,000 genes, and each one has a story of its own to be figured out, right?
Yeah, and so I'm actually, I could have told you from knowing a lot of the other parents that there doesn't seem to be any issues with the parents, but that does make it's kind of a puzzle then why it's so rare if nature seems to be so willing to tolerate losing a copy. Well, I guess that's genetics.
Yeah, exactly. So at what point did you decide to shift your career from cybersecurity research, computer science, full on into precision medicine? How did that coincide with the diagnosis and formation of the nonprofit organization as well, which I believe you founded?
Yeah, so we built a nonprofit, NGLY1.org, that raises money to do research into drug development for the disorder and to support the patients with the disease. It's funny. I think the conscious choice to change my career happened after it had changed in practice.
Right.
It was a gradual evolution, from one step at a time. So the first step was while I was still at the University of Utah as a professor of computer science, I picked up a second appointment in pharmaceutical chemistry and started collaborating with the folks there on drug development for this disease. Actually had some success. While I was at Utah, we found a couple different compounds that work for the disorder in a couple different ways, and then ended up on the faculty at the Harvard Medical School doing initially stuff more related to the diagnostic part of Bertrand's journey, trying to scale that part of up through the Undiagnosed Diseases Network, and then moving over to the therapeutic side there as well, doing that more bioinformatically, trying to use computation to predict treatments for patients. Whereas a lot of the work that was being done while I was at Utah was actually very, there's a lot of bench science involved, a lot of worms and flies and things like that. And then, in parallel with Harvard, I ended up getting an invitation to come to the White House. And I met with President Obama, ended up ultimately joining the White House itself, working as a strategist on the Precision Medicine Initiative, which was unveiled shortly after I met with President Obama the first time. And that's this program to collect 1 million genomes and medical records on 1 million Americans so that we can build what I call the Rosetta Stone of the human genome. So that was a very high level, entry point into precision medicine in terms of building the scientific foundation necessary to make it possible. Whereas the work I was doing at Harvard was more or on the ground with the patients. I also ended up co-founding a company called Paranomics that was doing repurposing drug screens for ion channel driven epilepsies. That company's since been acquired, but we did that very successfully. And so, at the end of this process, after 3 years, I think I spent 3 years working at the White House and 3 years at Harvard at that point, and I had been working in biotech. So I was on sort of indefinite sabbatical from Utah, and I realized, well, I guess my career has shifted, despite the fact that I still had an active computer science lab doing research and publications and grants and all that. Maybe I should consider going actually full time into medicine because I've been doing it anyway. And then I got so the plan at the time was to go to Harvard full time, but then I got a bombshell offer from UAB saying, will you come set up this Precision Medicine Institute?
Right.
Well, yeah, I guess I will.
You don't often get the opportunity to set up an institute, do you? I mean, I think it's unique, probably once, or if you're lucky, maybe twice in a lifetime, that they'll say, come and mold the institute in the way that you'd like it to be built.
It was an exceptionally rare offer, and rare in multiple ways, in the sense that, no, I'm not a traditional medical researcher. I don't have any formal training in the space. I've acquired a lot by, from trying to do this stuff for Bertrand and for others. But I think my formal training in biology ends with a C in sixth grade.
Right.
I'm not the typical profile of a medical institute director.
No, not at all. But sometimes that's good, right? Because it can shake things up. You bring a new perspective.
Yeah, and so I think UAB has ended up running a natural experiment in two axes. One is what happens if you put somebody from the patient side in charge of a research institute.
Right.
And the second is what happens if you just throw a computer scientist in that role too.
Right.
So both experiments are running in parallel.
Yeah, they managed to get both at once.
Yeah, it's been interesting. I mean, I think very successful as well.
Right. This is maybe a good segue into one of I tweeted out that I was going to have an interview with you and ask people if they had any questions. And one that came from Cecilia Lindgren at the Oxford Big Data Institute was to ask you about what changes in academic research you might, what one thing would you change about academic research and how would you go about implementing it? And I think given your experience from the government side, working on a biotech company and in academia and multiple different, wearing multiple different hats or in different fields, and now getting to start an institute from fresh, probably gives you a good perspective on that. So what do you think needs to be changed, and how are you doing that from your current role?
Yeah. So, I mean, the thing that comes to my mind is I would say, as a general policy in all directions, open by default. And by that, I mean, no closed data sets, no closed, code, make everything publicly available, from the get-go. And so, for example, for the primary piece of software that we built at the Institute, this AI tool that we frequently use for drug repurposing called Medi-Canron, it's open source, it's free, anybody can download it, anybody can use it. And that's the kind of philosophy I want to foster in all the research here. Coming from computer science, I noticed, a cultural difference over in biology and medicine where people guard their data and they guard their code.
Right.
And I think it should be just the opposite.
Yeah, absolutely. And it does seem to be a little bit of a tension, and it's hopefully changing for the better because there's more and more people in genomics and bioscience calling for open data, open code, and trying to, I think at the end of the day, accelerate progress, right? Because if it's locked behind closed doors for 2 years while somebody's waiting for a paper to come out, then it just slows everybody else down.
Absolutely. Yeah. I'm by no means the only one calling for this.
Yeah.
And I'm glad that there are many out there with the same attitude.
So how much of your research focuses on specifically ultra-rare epilepsies or specifically NGLY1, and how much is on precision medicine more generally? Because I know you also do work in cancer and common chronic conditions.
Yeah. So for the Institute as a whole, we have, 3 high-level focus areas. The first is rare disease sort of writ large, and within that there is an undiagnosed diseases program that's run by Bruce Korf, and then there's my lab, which does personalized therapeutics, and that actually extends beyond just rare genetic disease too. Right. Even if that's the primary focus or ends up being the primary client of that program, it's probably 80% folks with rare genetic disorders. The next most focus area is precision oncology. So Eddie Yang is my associate director there who heads up that. That's largely focused on genotype-guided treatments. And then Nita Lindy runs our pharmacogenetics program, where we're really interested in how do you actually integrate pharmacogenetics into a clinical environment,
right,
and make it useful. So that's in a nutshell. That's a very high level overview of the kinds of things we're doing here. I personally spend most of my time again on the personalized therapeutics front, where patients reach out. We will pull their data into our program, but program meaning the sort of a consulting program where we try to figure out how to map that data into the best possible treatment for them. And again, about 80% of those patients are rare genetic disorder patients.
Yeah, well, I think what's really interesting about that is if you think of the broad category of personalized medicine and what the term implies, that ultimately someday we'll have a personalized medicine for everyone, then at some point it kind of collides with the state of affairs today in ultra rare disorders. So if there's only 150 patients in the world with NGLY1, then the way we think about treating that disorder is possibly the same about how we think about a world where everyone gets the same treatment. So I guess in a way, ultra rare and common, but moving towards personalized medicine are not as far from one another as we might think.
No, they're really not. And I think, ultimately what we're doing is we're defining every disease into a rare disease, so that, we're heavily subtyping down to the specific molecular characteristics of every patient, so that every patient becomes their own instance of that disease. And so at that point, it almost directly overlaps with treating rare disease. So I certainly see it that way. Yeah.
Another question that we had on Twitter, which overlaps with this topic, is from Anna Mingorens, who works on a number of different rare disorders, but I know she works with Dravet syndrome patient organizations and CDKL5. And she wanted to get your opinion on the pay, as she calls it, the quote, pay $4 million for a therapy trajectory that we're headed towards in rare disease. So gene therapies and other very expensive but transformative drugs. How are we going to figure out these breakthrough treatments, but do it in an affordable way? And what do you see as the future here? Is it getting these costs down through technological development, or is drug repurposing maybe a lower cost way to do it? Or what do you think?
Yeah, in terms of whether or not it's going to be drug repurposing or these really customized therapeutics like gene therapies or antisense oligonucleotides, it's both. They both have an important role to play. And the good news is the cost is already coming down to a mere $2 million in some cases.
Right. So we're halfway there.
So yeah, I mean, these technological advances, do make a difference. And even when I was working with Paranomics, initially it was, it might have been $400,000 to do these repurposing screens at the start. By the time it was over, I think the cost was down to about $125,000 over the span of 2 to 3 years. I mean, it was not a long time, and the cost came down substantially. And I'm talking to folks now and think it could go further lower even still. So those technological gains are very real when it comes to driving down costs. And so I'm optimistic long term that will happen. And when I look at something like antisense oligonucleotides specifically, you look at the cost of just actually making the drug, like actual fabrication costs, and it's in some cases around $5,000 for the lifetime of the patient.
Right,
a lifetime supply of drug. I mean, that's amazing. So that to me, that says, well, that's where it's headed. $5,000 plus whatever it takes to design a drug that we believe is safe and effective. So it's the design cost that is really blocking us. And I think everything points in the direction of those costs falling.
Right.
I think specifically for both gene therapy and for antisense oligonucleotides, I think you will see an orthogonalization in things like gene therapy where you split out the vector from the payload, and you can sort of rigorously evaluate safety and efficacy of delivery and then in the vector itself apart from the genetic payload, and then we can have mechanisms for evaluating that as well. And I think a lot of this can be done computationally too. I think toxicities can be predicted in many cases in advance for things like ASOs. And so I do see these costs falling dramatically over the next several years.
Yeah, well, I was wondering if is it because they presumably will fall over the lifetime of patents, 10, 20 year time frame. But how about in the near future? I mean, we've had a couple of issues in the UK, for example, with the cystic fibrosis drug that took a very long time for the government and the drug maker to agree on a price. Do you think I mean, it's more complex than pointing the finger at one party, and I don't think it's helpful to do that. But do you think the pharmaceutical industry needs to focus on getting R&D costs down? Because I tend to hear most of the time the narrative is it's priced so high because the R&D is expensive, and to build a viable business, we have to recoup that. So is that kind of why you think the real driver is getting that R&D cost down?
Yeah, I mean, I think ultimately, yes, that, if you want to imagine that there's this perfectly competitive market for drug development, then it really is the cost of development that will drive the cost of the drugs ultimately. I don't know to what extent that's actually true. Right. In terms of the market for pharmaceuticals, but let's assume it is. Then you really do need to focus on several things. Actually, it's the cost of drug development, and it's the time scale, and it's the risk. So drug development is almost notoriously risky, and it takes a very, very long time. It is actually, in drug development, it can be difficult to just beat the cost of taking the same amount of money, sticking it in government bonds, and waiting 17 years. It's like that's what you're up against when it comes to making a profit. Like, you actually have to be able to beat your next best alternative as an investor, and given the time scales and the risks involved, that can be really hard to do. So I think the two things we can tweak there most readily from a policy perspective are risk and time scales. And getting drugs to market takes a very long time because clinical trials take a very, very long time. And I think there are ways that we could shift what we optimize in the process away from certainty and more towards value, because you can't really optimize both at the same time. And in fact, if you optimize certainty, you will certainly not optimize value for patients. That's absolutely the case. But that's the way regulatory bodies tend to behave.
Yeah, it's interesting. Can you unpack that a little more? So when you say certainty, I suppose you're referring to the clinical trial either passes or fails. You set this line in the sand, and everybody waits for the data to be unveiled, and stock prices go up or down, and patients' hopes are dashed. How would you re-architect that system in a better way? Would it be more continuous, or more, wouldn't be a discrete success or failure?
Absolutely. And certainly, approval would also be a function of the severity of the disorder you're intending to treat. So more severe diseases would require less certainty in efficacy, and in some cases, even less certainty in safety. I think for disorders that are, extremely severe, then we might even be willing to compromise a bit on safety, given the urgency of the situation. So I think that in general, I would say I would focus less on efficacy too, because I think the market could probably ultimately figure out a way to figure if the drug works or not.
Right.
Payers were certainly trying to figure that out as they do right now, and so I think that if you focus largely on safety and the known toxicities of a drug, you could chop off a lot of the uncertainty around and the timescale around drug development.
Is there anywhere where they're doing what you describe or thinking about? I mean, one thing it reminds me of is in the UK, at least for cancer, there's some discretionary funding that can be allocated to cancer drugs that are maybe not yet proven to be particularly cost effective or necessarily efficacious above the standard. So they haven't been cleared for everyone, but they can be kind of cleared in some cases. But did this come up in the Precision Medicine Initiative discussions in the US, for example, or are there any other healthcare systems where they're trying things like this, maybe faster clinical trials with more real world evaluation?
Well, so there wasn't a lot of that within the Precision Medicine Initiative. It really was more focused on enabling the science rather than changing the regulatory policy. Some of that was addressed in 21st Century Cures, but I don't think there's really been a substantive philosophical debate in the U.S. yet over how it is that we approve drugs. I think it's starting to happen, but I don't sense that we have the momentum for a sort of a full rethink of how we do it right now, and whether or not we're really ready for the truly personalized era in medicine. Because for me, we're ending up in absurd situations with repurposing where we are waiting for drugs to get approved for condition A so that we can take it off label for condition B. And that's a little bit weird. Like, we don't care if it's efficacious for condition A at all. All we care is that it's safe because we know from the molecular data it's likely to work for condition B.
Right. So there's some very clear, strange situations that are a result of some regulatory inefficiency and no other reason besides that.
Yeah, it's increasingly likely going forward that, when you're given a prescription, that it will be based on molecular characteristics that say this happens to be the right drug for you, and maybe nobody else with that condition. I mean, that could happen. And so the efficacy labeling is a little bit strange in that world. I think what we need there is, perhaps some trust in the processes themselves that end up making the predictions to say you should take this or you should take that. And then again, focus heavily on safety and toxicity. I think, regulatory bodies are I think pretty good at systematically characterizing the safety and toxicity of drugs. They can do that much faster than we can figure out efficacy.
Yeah, and I saw a presentation a couple weeks ago where it was the head of the genomics initiative at AstraZeneca, Carolina, and she was basically saying in the last 10 years, the reason that drugs fail has flipped. It used to be three quarters of the drug failures were due to safety, and now about 3/4 are due to efficacy. So things are generally safe now as they make it to clinical trials or past phase 2 clinical trials, but they're not. They're failing because they're not provably better than the alternatives, which is interesting and I think leads to your point.
Yeah, and what I worry is that in a future where we try to find a drug that happens to work for you, as opposed to a drug that was proven to work for the condition that you have, the drugs that didn't beat the effectiveness threshold and got lost for that probably had side effects that were beneficial in other disorders that will now never benefit from those drugs. So, again, I'm really very much against broad, large-scale efficacy as a condition for bringing a compound to market. I'm not against it in terms of payers demanding efficacy for paying for it. I mean, that's appropriate. But I also think we need to get to a different way of capturing the value of a drug. I mean, I think having payers pay only when the patient benefits, I think would change things considerably.
Yeah. Are you seeing, because there's a lot of discussion in the UK and the US and rest of the world about value-based care where we're only paying when things work, and I've heard mixed reviews that a lot of people talk about it, but it's very hard to implement. Are you seeing any traction as far as that goes from your work?
No, not yet, because it's really hard to define value.
Right.
So I say we should shift to value-based care, but then the question, what is value, pops up immediately. And it's hard to capture. And we see this in another form within precision medicine where we recommend an intervention, patient goes on the intervention, and then the question is, well, what does it mean to work?
Right.
And in some cases, it just smacks you in the face, and it's so obvious it's working. The patient has seizures and they stop. Okay, well, it probably was because of the drug, and there are ways to increase that confidence. But what if it's a condition where you're expecting to impact development? Well, what if it takes years now to figure out whether or not it worked? So it's really hard actually to answer some of these questions about the value of a drug. And so I agree philosophically that yes, we should switch to value-based reimbursement, but in practice it can actually be really challenging to define value.
How do you think we get there? There's, I think, some fundamental challenges that you describe where if something is not measurable in any realistic time frame, then maybe it's not possible to do it. But for other things, do you think we need more digital methods of monitoring patients in the real world and how they're improving and establishing baselines? Or do you think it's capturable through electronic health records or some combination of two?
Probably a combination. I think I'm very much in favor of more surrogate endpoints, biochemical endpoints to say, well, we've restored the missing enzyme, therefore,
right,
it's reasonable to believe that long term this patient will benefit from it. even if we don't see an immediate overnight impact.
What do you see as the biggest challenges in rare disease research specifically today? You've experienced firsthand the diagnostic odyssey. In my PhD, I worked on a couple of primarily diagnostics focused rare disease research projects, and this is, I think, still a huge challenge. But it's just the first hurdle for most families. If you have achieve that diagnosis, you start to think about improving quality of life, potential treatments or cures. Where do you see the biggest challenges and opportunities today in rare disease research, and where people should be focusing?
Yeah, so to be clear, diagnostics is still a challenge, even with all the tools we have today. I mean, if you look at undiagnosed diseases programs or the Undiagnosed Diseases Network, you're seeing diagnoses in about a third of the cases.
Yeah.
Even with the full power of genomics applied. And so to me, it says, well, there's a lot more to do. If we're only a third is great. It's a lot higher than we used to.
It's a lot higher than 1 in 10.
Yeah, no, we clearly have a long way to go, even there. And then, of course, once you get a diagnosis, there is almost no infrastructure at all today in terms of finding that best possible treatment, or any treatment, actually. And that's largely what I'm building at UAB with this institute, is the infrastructure to map every patient with their high-resolution molecular data over to the best possible treatment for them.
What kind of data do you use for that today? What are the main building blocks?
In the case of rare genetic disorders, the genome is the entry point, and it's starting off with the gene in question. And the initial thing we do for almost every patient that reaches out to us is impact analysis. So not just is this gene, or is this variant pathogenic or not, but what is the impact of this genotype on the function of the gene? And there's really 4 directions it can go. It's either overactive, it's underactive, it's absent, or it's toxic. So we right. branch in those directions, and depending on which direction we go, we either activate it, we inhibit it, we compensate for the absence of it, or we eliminate it. And we have strategies behind each of those too. So we're really rolling out this algorithm for constantly finding next steps in this therapeutic odyssey.
So who's involved if you walk through kind of a typical patient journey through something like this? Are they interfacing with a medical doctor or a data scientist, or is it a team of multiple people? Because it seems like a very interdisciplinary thing.
Yeah, it's a very interdisciplinary team. It's believe it or not, a lot of the heavy lifting is done by undergraduates that are trained to serve as the intermediaries between the patients, the scientists, the artificial intelligence tools, and trying to coordinate everything all together. And ultimately, if it succeeds, it results in a letter from us to the patient's physician documenting research findings that may be applicable to their particular patient. And then we leave it in their hands to make a decision as to whether or not they want to change care based on those research findings.
And so I guess it's very
highly interdisciplinary, with undergrads serving as a sort of glue.
Right. And I guess it must be very dependent on the physician on the receiving end of that recommendation also being invested in the process, right? Because you hate to do all of that work and then have it lose steam at that point. So do you spend time speaking with the patient's physician or their clinical geneticists or other specialists to try to make sure that it's well received?
We do. In fact, we consider that part of our research. So we're doing, we're analyzing the rate at which recommendations are adopted, and we're even doing essentially user experience studies on the format of the letters we send themselves. So we're moving to a format now where we have the really immediately relevant stuff out in boxes at the front. So you can glance at the first page and say, oh, that's what this letter is about.
That's amazing. I'm not aware of any program like that. There are elements, I think, that you've probably drawn inspiration from other places, but that whole package together is really fascinating.
Yeah, well, but you're right that we do it because it really sucks if you spend a lot of time, and we could spend dozens of hours or even hundreds of hours on a single case in some cases, and we'd really like to make sure that the physician reads the letter and actually does something with it in that case.
How many patients are you able to get through this process in a typical year?
So in the past year, I think we've had engagements with about 300 patients.
Wow.
That seems so. Yeah, young program. It's only been around for about a year and a half, but I'm guessing that's going to be roughly typical. It does seem to be accelerating. We do seem to be able to handle that level. A lot of the work happens at the initial engagement because it goes to impact analysis and the formation of an initial plan. And then it will oftentimes wait for a while because some of these next steps can take a long time.
Right.
So you can have a lot of cases moving in parallel, waiting for an update.
So you have a team of undergraduates. What an amazing undergraduate experience as well to be coordinating something like that.
It is actually, and oddly enough, all the undergraduates that have been through this program, I think 5 are now alumni of the program. All 5 have gone to grad school,
right.
med school, or to get their PhD. So I feel like there might be something to it. Actually, I think it might be a way of really energizing undergraduates in a way that makes them want to go on and pursue medicine or science for their career.
Yeah, well, I think maybe part of it is that actually putting people at the interface with the patients and not, at a cell culture helping cells grow or dealing with mice where you're very far removed from who the research is designed to help. Obviously, we have to have that kind of research. I sat behind a computer for my whole PhD and didn't really interact with patients, but having that patient-facing experience, even if you go on to just do lab work for your PhD, for example, it gives you that context, I think.
Well, in fact, this just happened. One of my students, she's a PhD student. She's like, what? I realize I need to go into the lab now. So she's going into a bioinformatics lab that's also it does a lot of bench science. She's like, I know a specific problem I have to solve in order to help patients.
Right.
So she's going and spending hours at the bench now.
That's great. So if you look forward 5 years, when did you come into the job at UAB? Was it about 1.5 years or 2 years ago?
2.5 years ago. And then this program for patients started around 1.5 years ago.
Right. And how far are you through the institute building process? Have you hired all of your faculty members, or are you still in the process of building that team?
Oh, we are definitely still hiring faculty.
Right.
So we've hired a number. I think we've probably hired 5, maybe 6 faculty in the past 2.5 years, and we'll probably hire as many in the next 2.5 years. There's definitely a broad vision of the kinds of science we need to work as a team to produce these solutions for patients. And we're definitely doing targeted hires in those areas.
What is that vision? If you look forward 10, 15 years, whatever the horizon that you have is, what you all want to try to build or see in the world?
So the vision is to have a scalable resource for patients and physicians where they can reach out when they hit that challenging case that fails standard of care and needs that truly personalized care, so that we can, within a reasonable amount of time for a reasonable number of patients, return that report to say, here's your option. I'd love it if, for maybe 80% of the time within 2 or 3 months, we return a report that says, this is what you do.
Right. That's amazing. No, I think, and it's sorely needed, especially use the word scalable, but the idea that you can start small and figure things out, but build it in such a way that when something works, you can really bring it to thousands or eventually millions of people is super important.
Yeah. And that's why the operating philosophy of the Institute is anytime we do something manually more than 3 or 4 times, it has to get automated.
Right. Right.
That's just drawing on my background in computer science. Like, I just don't like to do stuff that can be automated. And so I think that's where the scale is coming from over time, and we always automate the most recent bottleneck.
Right. Now it's I think a lesson that cuts across all industries, isn't it? If you're starting to do things too many times, you need to think about how to streamline and automate it.
Yep.
So I know we're running out of time here, and thank you so much for taking the time to do this. I was just wondering if maybe as a final question, if you put yourself back in your own shoes, and I suppose it was probably 8 or 9 years ago at this point when you got your son's diagnosis and you formed the nonprofit for parents that are maybe in that position right now, where they've recently had a diagnosis or they have a child that's experiencing some kind of unexplained symptoms, do you have any recommendation of what you would have told yourself or what you'd tell someone now on how to best set themselves up for success in terms of early diagnosis, getting into research?
Yes.
Okay, good.
I definitely have some advice for my old self.
Right.
So I think the one piece of advice I'd give is be careful about over-investing in the basic science.
Right.
If your goal is to find a treatment, you can spend forever working on the basic science and get nowhere. You really want to focus on the translational science around a disorder. And the first insight I have to share there is that you do not have to understand a disorder in order to treat a disorder. And this runs contrary to what every parent thinks. I think initially is like, how can you treat a disorder without understanding it? And what I've come to realize is that what you really need to do is get as fast as possible to what I would call a screenable model of the disease. And that could be a cellular model. It could come from the patient themselves. It could be a model that you build in some way with transfection. It could be a fly. It can be a worm. It could be a zebrafish. But you need to find a model of the disease in some system that is amenable to drug screening. And once you have a model that has a phenotype that you can recognize in some way, you want to test every approved drug in the world against it. That's basically it.
Right. I think that's super useful advice. So you can think about all of the pieces you need to get in place to get there. But if that is the core piece, then you can think about raising research funding or finding an academic that will help you do it, or finding a contract research organization or partnering with a company. There's a ton of ways to actually achieve that. But the key is actually to get to that screenable organism.
Yeah, and there are examples now that I can cite. So Ethan Perlstein's work in the glycosylation disorders community, where he was building animal models like flies and worms and yeast, and just doing drug screens to see what comes up. Where for a tiny fraction of the amount invested in the basic science of these disorders, he actually went all the way to treatment recommendations in a much shorter amount of time. And we're talking about the time scale of a year versus decades, and hundreds of thousands of dollars versus millions of dollars spent in doing that work. And a lot of the parents that begin the journey that we did, and I was the same. I invested in the basic science, and we learned a lot that way. We really did. And I'm not sure that much of it has been essential in actually finding treatments for the disease. Some of it has certainly helped, but most of it has actually come from finding that model and interrogating the model.
And I suppose sometimes it might work the opposite direction, where if you find something in a translational in a translational research program, then it often can tell you something about the basic biology. If you find a drug that works and you figure out what that drug's doing, then it probably will tell you something about the biology, right? So you can potentially achieve some of that early understanding anyways.
Oh, that's absolutely true. And it's certainly happened with my son's disorder, NGLY1, where we found stuff, saw that it was working, and then went back to figure out all the particulars of why.
Right. I remember reading in your blog posts, you tried a couple different things. You've tried ketogenic diet. You've tried a few different anti-epileptics. Is that right?
Right. Well, that was all pre-diagnosis too.
Right. Right.
So we found some stuff that helped pre-diagnosis, and some of it we have a better understanding of why that stuff may have helped. And since then, we found a number of things that also start to chip away at the phenotype.
Right. Help you really understand what's going on. I mean, it's such a complex system, right? We can't, like you said, we can't hope to understand exactly what's going on. Sometimes we have to just learn by experimenting.
Yeah. I mean, I would say that, when it comes to drug development, finding a model and then doing trial and error, just trying lots of different stuff on the model is remarkably effective.
Right. No, that's great. Well, I appreciate you giving that advice. I think it's always good to have someone who's been through this incredible career trajectory to just say, if I could take a deep breath and go back and do it again, here's what I'd change.
Yeah. So the second piece of advice would be get a board of advisors or scientific advisors that don't have a conflict of interest. That's also pretty important.
Is that speaking from personal experience, or have you seen other
that's from the experience of others.
Right.
When you start a foundation, the scientific advisory board that reviews the grants should not be able to receive the grants.
Yes.
To put it simply.
Good. Very good advice. Are there any organizations that you recommend that help patients get started forming patient organizations? Here in the UK, in Cambridge, there's Find a Cure, which is a very good one.
Right.
But are there others in the US or elsewhere in the world?
So like Global Genes here in the US.
Right.
That seems helpful in this regard. I think Nord may help with some of this too. I think Nord and Global Genes are certainly two of the big ones.
Great. Well, I just want to say thank you, Matt. I really appreciate you taking the time. You're on a lot of our lists of great advocates for rare disease and examples of people that just don't take no for an answer. And when there's a problem, they push right through it rather than throwing their hands up. So thanks for taking the time to do the podcast. I think people will really appreciate it.
Well, thank you. It's been a pleasure.